Thank you, Jean. And thank you all for staying to the end. Hope you enjoyed the refreshments. So I think I might have oversold the talk a little bit when I look at the title, but I will do my best to highlight how understanding basic virus evolution can hopefully help you towards designing better vaccines and also future therapeutics. But of course, because it's an inaugural lecture, apparently you have to give a little bit of background about yourself, which is something I never really like to do. I'd rather people don't know me at all, in truth, but there you go. But I thought I'd better dedicate the talk to my parents, and the reason being that when I got my chair at Nottingham, I was previously at Nottingham, it was the same year that my dad died, and so it got cancelled. And so I thought, well, you know, and everybody said, when are you going to have your inaugural lecture? I'll have it soon, and it never happened. And unfortunately, this year my mother died, so the moral of the story is don't agree to an inaugural lecture, because horrible things will happen. But as I say, this is dedicated, I guess, to their memory. And so what I'm going to hopefully cover is a chronology of my life, as poor as it's been, and really just to hopefully give a highlight of the journey that I've been on, which has been odd in terms of I don't think any of us really ever have any idea where we will be. Certainly if you'd have asked me 3 years ago, I wouldn't have said I'd be in Liverpool, for example. So things happen and you go with it, and hopefully you enjoy it on the way. So I'm going to start at the outset, where my upbringing, then my journey to Bristol, and then onwards to Brum, brilliant curries, and then the return to Nottingham, and then finally whether I was going to turn red or blue when I came to Liverpool. In fairness, I was probably already red, but there you go. So that's the village that I spent my childhood. Tallest pit headstocks in Europe, second tallest in the world. There's the first fact. They're still standing, so there's a preservation order. So even though the pit closed in 1994, the wheels are still there, and in strong winds they still turn. Okay, but really that kind of set my childhood. It was a great village to live in. There was a great sense of community. Maybe it wasn't the most academic of environments, but we had a lot of fun. That's all I can say. So I went to that school, Garibaldi School. As I say, we had a very enjoyable time, but it was a fairly small sixth form that I joined, so I didn't leave school at 16 as I could. I stayed on into the sixth form because I wasn't sure what I was going to do. And then I got into the lower sixth, and it was Christmas time. So hence, have I got this right? So hence it's not working with the red laser anymore. And what had happened is two of the teachers that I had, maths teacher and the form tutor, said, what are you going to do with your life? Which I thought was a bit heavy. You know, it's Christmas. So then they said, well, do you want to come and choose a Christmas tree? And I thought, well, I can cope with that. Okay. And so off we went to the Forestry Commission to choose a Christmas tree for the school. And then when I got back into the bus, they just basically refused to move until I said what I was going to do with my life. So I said, well, I've not really thought. You know, I've not really got any plans. And I think the options were pretty much, I could follow my dad, who's there. I bet you can't recognise him. He's the one with the smiley face. Okay, so I could follow him and my other brother down the pit. But I think if I'd have wanted to do that, I would have done that when I could, the year before. Or I could go building, because I quite enjoyed that. I did some summer work at a local builder's. It's very shoddy brickwork, by the way. I wouldn't employ him. But again, the thing that put me off was winter. Picking up bricks in winters, you know, and they stick to you because of the ice, that's not very good. Or, quote, you could go to college. And that's what we called going to sort of higher education in those days. And so that's what I did. I'd missed UCAS, so I couldn't apply to any universities. I probably wouldn't have got into any anyway. So I applied to Bristol Poly. I'm not sure why, I just like the look of it. And actually Bristol's very much like Liverpool. So that's where I went to. It wasn't, I can't remember it looking like that, but there you go. Maybe that's just some PR. This is what they do in comms, don't they, Marie? Okay. And again, I wasn't really sure what I wanted to do through the undergraduate degree until I had a couple of lectures and a couple of practicals with a virologist called David Shaw, and I still keep in touch with him. He's very old now, excuse me. Brilliant organist. He's also a great composer, so he plays in the local church. So he's a gifted guy. And I started growing herpes viruses. And I thought, this is pretty neat. I can't see it, you can't see anything, but you can see the consequences of this herpes virus. And at the time, he'd just discovered that caffeine and theophylline, which is present in tea, can inhibit herpes viruses. And so I thought, that's pretty neat. And particularly neat because we didn't read it in a journal, we heard it on Radio 1's Newsbeat. So that was even more engaging. That's science. Forget Journal of Virology. This is where I want to be. And so really the job was done. I decided at that point that really I was going to be a virologist. So what is a virus? So if I was to ask Nick, if Nick's here, Nick Casewell, apparently it's a medieval term for snake venom. Is that right? I don't know. But it's an infectious agent, very small size, simple composition, but they basically are what we call obligate parasites. So they have to get into a living cell to be able to replicate and then to produce new viruses. So we have viruses of animals, plants, bacteria. Basically, if there's a life form out there, you can guarantee there'll be a virus that infects it. And it's essentially that the word is derived from a Latin word meaning slimy liquid or poison. And we have sort of two flavors of viruses. Well, we have lots of flavors, but basically you can have a naked virus, which you've got on the left-hand side. So basically it's a protein shell with a little bit of what we call nucleic acid, so genetic material on the inside. But we also have slightly more complicated ones called an envelope virus, and these are the ones that I generally worked with for the rest of my life. So these have a layer of lipids, a layer of fat, and embedded within that lipid membrane are proteins that allow the virus to get into the cell. And those are the targets of your host antibodies. So we've all heard of the spike protein because of the COVID outbreak. So basically those little projections are the spike proteins. And lots of these envelope viruses have them, and they use them to get into the cell. What have I done? Broken it. But not all viruses are the same. And I think what I want to really point out here is the genetic material that viruses contain. We've got DNA in our cells. DNA is very stable. It means that we can have very big genomes. But lots of viruses have an RNA genome, and their genomes are very, very small. So the amount of genetic material that they have to each individual is tiny in comparison to us. And so we can see there a selection of RNA viruses, rhinoviruses, which cause colds, rhino, nose, measles virus, polio viruses. So these are all viruses that we've heard of. And because they've got an RNA genome, they can evolve incredibly rapidly. And, whoops, if I go back there. So you can see on the left-hand side, this is essentially a measure of how quick a virus can evolve. And you can see that the RNA viruses can evolve at a rate about 1 million times faster than you or I can. Not only that, they have a very short reproductive time. They can reproduce within hours, whereas as opposed to us, sort of decades. And so that means you can have a very diverse population of viruses very rapidly. And this gives them an evolutionary advantage. So what are the consequences of virus evolution? Well, it can change what we call their tropism, their ability to infect different tissues in the body, or their ability to infect different animals. So, and really the first barrier for a virus to make a jump into a new species is that cell entry process. Can the virus get into the cell to replicate? If it can't, it's a dead end. And so we've been very interested in this cell entry event. In fact, that's all I've been doing for the past 30 years. Very sad, I know, but it is important in terms of what the virus life cycle, where that's concerned. Okay, so this is what happened during the Ebola outbreak in 2013 to 2015. So you can see the affected areas, Guinea, Sierra Leone. The epicenter, as it were, was this part in Guinea, And you can see the virus starting to spread locally, but also into Sierra Leone, into Liberia, and then that virus is exported back into Guinea. So this is the time interval here. And here at the top, you've got a family tree of the viruses. So this is the virus that spilt over into the first humans. And then you had lots of human-to-human transmission. All the time the virus was changing, evolving, adapting, spreading. And so eventually, by the end of the outbreak, you had viruses which were very genetically diverse from the original virus. It had changed as it had spread from human to human and through different geographical locations. And you can see the numbers of viral infections spiking at this point, about mid-2014. And then eventually that outbreak is brought to an end because we did some very basic infection control. We didn't control it with vaccines or treatments. It was going out, safe burials, education, and good infection control. So that got us thinking, and Rich, who did a lot of this work, is just sitting behind Joe Clegg. So be careful, Joe, he's not to be trusted. So we asked the question, with all of this human-to-human transmission and all this genetic change, had the virus evolved? And at the time, it was a bit of an odd thing to suggest, even though we know viruses evolve quickly, but a lot of very established people in the field said that's nonsense. Keeping an eye on time. And so this was the fundamental question. We knew that the glycoprotein in the surface of the virus had kind of changed from green to blue. It had evolved, it had changed properties, or we thought it might have changed properties. And so how do we study whether or not these viruses or these glycoproteins have changed the way that the proteins behave? Well, we can trick HIV particles into incorporating these surface proteins into their own shell. And so we can measure the ability of these proteins to mediate entry by bolting them onto HIV particles. And inside those HIV particles, we've got a reporter gene. It basically emits light. It literally is a light-emitting gene. So if we put those viruses into some cells and they can replicate, for example, in bat cells, we get light. If they can't, it's dark. Okay? If we put them into human cells, same thing. If they infect, we get lots of light. If they don't, it's dark. I'm probably underselling virology here, but it is that simple. Okay, so how do we do it? We basically have 3 different pieces of DNA. One piece of DNA encodes for the HIV viral particle, which is the red bit there. Another piece of DNA encodes for that reporter gene, the light. And then we can bolt on lots of different envelope proteins from a whole variety of different viruses. We can use Ebola, Lassa fever, SARS coronavirus, Nipah, I could go on endlessly. And that way we can generate particles which tell us how the virus of interest enters cells. And so we literally ask the fundamental question, if we take these viruses that have evolved and try and infect bat cells or human cells, and for a while we wondered if we had to hang the bat cells upside down in the incubator, but apparently you don't, so that was good. We could just use a normal flask. And essentially we were looking for either high infection or low infection. And so remember the tree that I showed you. Essentially, here's the early viruses that emerged in Guinea, probably from a bat, into humans, and then they transmitted and evolved, and the glycoprotein, the surface protein, evolved and changed. We're able to measure those different viruses. And hopefully you can see here, that's the initial Guinea virus, and that it was that much infectious in humans. But as we saw the epidemic continue, you can see that the viruses, without doubt, evolved. They became more fit for human infection. It's probably the only bit of data that I've ever seen Rich collect where we didn't even have to analyze it. You could see it on the machine and you could just see that, yes, the virus has been evolving as it's been spreading through human populations. It's obvious to us now, but at the time, people didn't think there was enough time for it to evolve. And you can see, maybe less strikingly, but here's the infection of the virus at the outset in bats. And then as the virus was spread through humans, you can see that that started to decrease. And that's classic virology. It's how we make vaccines. You take a virus from a human and you grow it in animals, and it becomes what we call attenuated. It becomes less able to replicate in humans, and then we can use it to provoke immunity without causing disease. So the answers were already out there, but people just doubted if it really happened in reality. Okay, so that's virus evolution. But one of the things that virus evolution allows a virus to do, keep my eye on time, is immune escape. And so when I moved to, a lot of the early work that I'd done was at Porton Down in Birmingham, but then when I moved to Nottingham, it was a group that focused mainly on hepatitis C. And if we look at the problem with hepatitis C and immune escape or genetic diversity, we've got 3 family trees here. So the length of the branches is the amount of genetic diversity. So long branches, lots of genetic change, and therefore difficult to generate vaccines which will protect against them all. I managed to lock my thing on that now. How do I get it off? There you go. Ooh, well done. There we go. Right. So you can see here's hepatitis B, not much genetic variability. HIV, a little bit more. Now we know how challenging that is in terms of vaccine design. We haven't got a vaccine for HIV. This is hepatitis C by comparison. The scale is exactly the same, but so much more genetic diversity. And the other problem we've got with trying to develop vaccines and treatments is that very often viruses hide the important parts of their glycoproteins, the bits that you really want to target for a vaccine or a therapeutic antibody, particularly through use of sugars. So they literally coat the surface with sugars, and your immune system doesn't recognize the sugars because basically you're very sugary. Most of you are very sweet, but you didn't realize that. Remember, next time you have a fallout at work, think glycans, sweet. Okay, but also occluded epitopes. These are hidden epitopes. This is one of those posh cauliflowers, and I can't remember the name of it. Anybody help me out? Florietta or something? Anyway, posh collie, okay? And this is a brilliant illustration for what a viral glycoprotein looks like. So you can see that there's a lot of surface which is accessible to antibodies. But you can also see there are little pockets in there that are going to be a lot more difficult for an antibody to get at. So what we're trying to do, and what viruses do, is, and I'll come to this later, is change a lot of this surface so that it escapes antibodies, but keeping these hidden bits the same. Because what it does is use these hidden bits to do things like lock onto cells or to drive part of the entry process. So the challenge is, can we identify regions of the virus which are conserved that we could target with antibodies or target with vaccines? So the first question we asked with hepatitis C, was do cross-reactive antibodies really exist? And so here what we've got is on the right-hand side, you've got different genotypes of hep C. Remember the tree, we're taking little viruses sampled across that tree, so it represents the genetic diversity. And here we've got sera from people who are infected with different genotypes of hep C. And so what we ask here is, how reactive are those sera to those proteins? So if you've got green, it's good. Okay, so you can see here, you've got quite a lot of green. So we know that the cross-reactive antibodies exist and they can cross-react with all these different strains of virus. But this is against protein as it exists on the surface of the cell, of the virus. So it's a bit like that. It's a bit like my lanyard at the moment, so properly folded. But if we denature it, basically boil it like you do an egg, and then ask the same question, how much conservation is there, how much cross-reactivity, I'm hoping you'll see that most of that green has now disappeared. We still get some green maintained, and this represents cross-reactivity with viruses that we know are easily neutralized. But unfortunately, not that many people are infected by those sorts of viruses. Most people are infected by these viruses that are a lot more difficult to target. So the question that we have to ask ourselves is, can we identify the antibodies that recognize these conserved epitopes? So see this patient here, you've got good green all the way down this panel. What is it in their sera? What antibody is it in their sera that gives it that good breadth of activity? And so what we essentially did was again use the pseudotype assay. I told you it's a common tool. We use it every day. So instead of bolting on Ebola glycoproteins, we bolt on hepatitis C glycoproteins, but it's the same sort of assay. And we try and identify those sera or those antibodies which can kill virus. So here you've got infection of the virus, and as you increase the amount of antibody, you can see kill. Here, you can't see much killing at all. Here, you see resistance killing. So we're trying to focus on these patients here and what is it within their sera, which is giving that good virus killing. And so we were able to tap up, for want of a better word, researchers around the world who've been developing monoclonal antibodies themselves, but they're all sitting in different labs around the world and nobody's doing anything with them. And so what we did was we contacted them all, said, please send us your MABs, we'll analyse them and we'll tell you how good they are. And so we did that, and we identified a number of monoclonal antibodies. So these are single antibodies with a single specificity, and hopefully you can see that this again is lots of different virus strains, and some antibodies give good reactivity across the whole panel. There's still some viruses maybe they don't miss, but you'll never get perfection. Okay, but we're trying for that breadth. And we're also able to show that some of those monoclonal antibodies could protect mice. Well, look how I've spelled protect. I was having a moment. Okay, but they can protect mice. So here you've got infection of mice over time. So this is a poor antibody, and you can see that it doesn't really protect the mice. They get infected. Here you've got an antibody that gives a little bit of protection. But here you've got an antibody that, on the whole, protects most of the mice. So we know that there are antibodies out there. So we'd identified a limited number of antibodies which could kill lots of different virus strains. And so we embarked upon a fairly large project to take some of these antibodies into clinical trial. So we had partners who could make the antibody to GMP standards, so to good standard, clean standard that you could stick in a human. We had people that could test the antibodies, had people who were interested in the kind of commercialization of those. So we're all ready. And we got to the point of clinical trial in Barcelona, and then this happened. Sod's law. And literally this was about 10 years worth of work. And so suddenly we had no patients to do the trial in, because suddenly there weren't so many people needing a liver transplant, and the liver transplant individuals could be safely treated as well. Now actually we're revisiting this project, and that's for prevention of infection in particular cohorts who would keep getting recurrent infections for hepatitis C. But this essentially stalled that work. And so I was at a loss as to what to do next. So I did something entirely different, which is what you do. So I started to engage with the media a little bit more. So I'm just going to break it up a bit. There's a bit more, too many antibodies at the moment. So I just want to give you a little sense of my experiences with engaging with the media. David Heyman, he was important with the initial SARS coronavirus outbreak. He headed the WHO response to that. He's a very nice guy, very sensible guy. I'm sure many of you know him. Bertie's nodding his head. Done a lot in terms of global health. But essentially, he'd said, you know, if you don't engage with the media, and we heard it as again with the symposium, you know, misinformation is a very, very dangerous thing. And so we really do need to engage and take this seriously. So what's my own journey? So, excuse me, for a while I'd been engaged with the Science Media Centre, writing comments on particular stories. So they send you a press release, you send them something back, and then they'll either use it or they'll bin it. But that's fairly easy to do. It's fairly anonymous as well. But I always avoided broadcast. It was a very difficult thing for me to engage in. I started to do a bit of local radio, particularly if it was Radio Norfolk, aha, Alan Partridge. Mainly because I thought nobody will know me from Norfolk, and that has remained the truth to this day. Okay? But Radio Nottingham, that was slightly different, you know, because you knew people. There was a chance that they would have heard you. Okay, so I really didn't like to do that. And I certainly wasn't ever going to do TV. And the problem was that because of the stuff that I worked in, whether it's emerging viruses or HIV, there would always be something coming into your inbox about, you know, will you go on the whatever breakfast sofa or the sofa with Philip Schofield? Glad I didn't do that, as it turned out. And my reply was always, I didn't reply. And you can find 100 reasons why you don't engage with them. There's always somebody better out there, more expertise. You're always worried what people might say in the corridors. Okay. Yeah. You can make yourself look a right idiot. I've done that. You have to move on from that, though. And also this wrong impression that journalists are out there to catch you, to trip you up, to sensationalise. I've rarely found that. There are certain outlets, I'm sure you know who they are. And then into my same inbox came an enticing invitation to apply for a British Science Association Media Fellowship. And they ran over the summer. And I thought, why not? Because I knew inwardly I really wanted to do it, but I just didn't have that confidence to go the kind of next step. So I was very lucky. I got a fellowship to go and work with the BBC down at London, New Broadcasting House, to spend a few weeks with the guys who did news gathering, science writing online, great guys. Then I was to go away to the Science Festival, which I think is this year in Liverpool, to report back for the BBC website, and then back to Broadcasting House to help with some radio. So I was really looking forward to it. And yes, everybody's question, I did. Okay. That's the only thing people are interested in. Did you meet Brian Cox? Why? And we even wrote a story about him, myself and Nick Crumpton, my partner in crime. And he was he was okay. Right. And actually that experience of spending time with the BBC really changed my attitude to media and engagement. And I ended up going back to the BBC every year up until the pandemic because it was different. I enjoyed it. And after a while, they kind of started to trust me. So I know sometimes if people were on holiday, they'd phone me up and say, do you fancy coming and do a week online? And yeah, why not? I'll go and do that. So Nottingham were very good to me like that. So, but of course, it meant that Nottingham's, it was known for hopefully the right thing. So I continue to work with the BBC, not so much now, but I did up to the time I left Nottingham. Yes, I've done feature writing. The Daily Mail wasn't a great experience. It is out there. Please look at the comments. And in fact, I did develop a love of media, and especially radio. I loved radio. It's a fantastic media. And indeed, got the chance to present a mini-series on Radio 4, which was really enjoyable. Unfortunately, the COVID pandemic came along, so that was the end of my sort of radio sojourn, because then the science unit moved to Cardiff, and working with Cardiff is just more difficult than working with London. But anyway, back to the story, as it were. I still refuse the breakfast sofa, so much so that I've upset all the producers in Manchester. But that's not a bad thing, is it? Okay. So remember, please, this is a kind of ask for all of you out there to try and engage with the media. Register yourself on the Science Media Centre if you're not already. They do media courses as well. Please sign up to that if you get the chance. Sorry, David, I'm not telling everybody to do things that we're not sanctioning here, but it is important because we heard about misinformation. It's a serious thing. And, just keep practicing. Think about your audience. Don't get bogged down in detail. Sound bites. All outlets love sound bites. It's not trite. You can still get a powerful message across, but you just don't need to bore somebody with the intricacies of viral entry like I did do in the first part. It can be very demanding. That was my wife's birthday, and I had something like 100 requests come in because it was like right at the peak of the Ebola stuff, and because it was early days, I thought I'd have the energy to kind of do them all. But once you do engage, then you have to keep engaging. They soon fall out of love with you if you start to refuse. But it is, here we go, I did this for Marie, but it is rewarding, okay? So the collective voice of scientists can put pressure on politicians. It definitely helps spread facts, not fiction. And if you don't help the hack, somebody else will, and that might not be the right message. And of course, you may get your own bus, which I believe is still running on the 35 line. That was quite horrendous when I first saw that. It came as a bit of a shock, but there you go. Right, so back to the science for the last 10 minutes. So preparing for the next pandemic, and some of you who are at the symposium have heard a lot of this, so I apologise. But think of it as a revision session, Okay, there will be questions at the end. So can we use the sort of tools that we generated for hepatitis C and for the other viruses? Could we develop the understanding about what therapeutic antibodies might look like to develop therapeutics for the next pandemic? Remember, we'd had that kind of stop of Hep C research. We're in a cul-de-sac. Where to next? Excuse me. And so I'm going to talk a little bit about some antibody discovery, particularly against these bat coronaviruses, which circulate predominantly in, a lot of them have been found in China, but I think that's mainly because that's where a lot of the virus hunting has been carried out. These viruses will exist almost anywhere and everywhere. Coronaviruses are highly prevalent in bats, and bats are found on every continent. So you can fill in the missing bit. But back in 2015, Ralph Barrick and his team had described the fact that there was a whole cluster of viruses infecting bats that were very much like the original SARS coronavirus of 2002. So back in 2015, he predicted that we would have a coronavirus outbreak. This was then later picked up by the conspiracy theorists to say that it must have been a lab leak or an engineered virus because he'd already done it. Okay, nonsense. These viruses were out there. They were just waiting for the opportunity to emerge. And in his paper, he showed quite clearly that these viruses could infect human cells pretty much as well as they could infect bat cells. And unfortunately, in terms of antibodies, we had some antibodies that might be able to kill them, but we had most antibodies were unable to touch them. And so we were interested in, can we have a pan-coronavirus or pan-sarbecoviruses, as this group of coronaviruses were called? We put in a grant to MRC to develop cow antibodies, because cow antibodies are quite unusual. And at the time when we put the grant in, this paper appeared in Nature. And we thought, damn, we're sunk, because somebody's already done it. Actually, this was a huge benefit. Because somebody had done it and proven it with HIV, it meant that our grant flew through grant review, because they said, of course it's possible. Brilliant idea. The Americans have done it, okay? So again, fickle finger of fate, but this time blessed us. And so what is it special about cow antibodies? The special thing about cow antibodies is about 10% of their antibodies have this extension which can access into these hidden pockets on a virus surface. So if you remember, the pockets that we're interested in, well, basically deep pockets are no match for cows, okay? So if we think about what an antibody, for some reason, doesn't matter. There's a few slides missing, but we won't worry about it. So essentially, what happens with humans is very often the antibodies that we're interested in bind around this pocket. And so they block this pocket, but they don't physically interact with it. So what does the virus do? It just changes some of these parts, changes colour, changes charge, doesn't change colour, but it changes charge or changes shape, and the antibodies can't bind anymore. But the pocket itself remains protected. So if this was a receptor binding pocket where the virus latches on, see, there's a receptor. You use whatever's available, okay? There's a receptor, there's the pocket. It can still engage with that receptor. Okay, so your antibodies really struggle to keep up with the evolution of the virus. But we thought the cows will sort this out, because surely they can produce antibodies which can get into these occluded epitopes. So we got our two cows, Jack and Dougal. We'd been working with these cows, mainly on Lassa fever virus antibodies, but COVID came along and we thought, let's have a look at some pan-coronavirus. So what did we do? We took the proteins from the surface of the coronavirus. This was SARS-2. This is a cartoon of what that glycoprotein looks like. It's got sort of 3 subunits, so it sits like that. They're identical units, but they come together to form a structure with 3 subunits. And that spike protein interacts with the cell receptor called ACE2. So that's what the virus uses to get into the cell that it's infecting. So we took different versions of this spike protein and immunized Jack and Dougal. So we took S1, which is just this part of it, so like a truncated bit here. We then added what we call full-length spike, which isn't quite full length, but it's all of this bit which sits on the outside of the virus. And then right at the end, we took a trimeric version of it, so the 3 subunits together. So the cows had seen S1, S1, S1. We gave it a little flavour of full length, and then we gave them the trimer, and then we bled them to see what they'd produced. And then what did we do? This is as technical as I get. We took the blood. Okay? We layer it onto a sugar solution, and then when we spin it in a centrifuge really fast, what that sugar solution does is it's able to separate out the white cells, and it's the white cells that contain the B cells, and it's the B cells that produce the antibodies. So we've got a source of cells that can produce antibodies. Oh, and the other thing that we can do is we can take the plasma off the top, and we can see what antibodies the cow was producing when we bled them. So we're able to take that plasma and then test it for its ability to kill virus. Do we suspect that those B cells contain antibodies that we're interested in, is essentially the question. And so when we did that, we could see that the cows had indeed produced antibodies that could kill, so neutralize the virus. So 100% neutralization, virus wiped out. Then as we dilute out the plasma, you can see that the virus starts to infect and replicate as normal. But the key was that we could kill all of common variants of SARS-2, but we could also kill the original SARS coronavirus that emerged in 2002 that the cows had never seen. No human had seen since 2002. So how do we then identify the B cells in the mixture of white cells that are in that sample? I'm saying it's easy, but as Rich knows, it took us a long time to develop this, but theoretically it's easy. So essentially you take the spike protein of the coronavirus, you label it with 2 different colours, literally green and red. They fluoresce when you shine a laser on them. And then if you mix that spike protein with the B cells, they will label the B cells according to the type of antibody they've got on the surface. If they've got an antibody on the surface that doesn't interact with spike, they won't bind. If you react them with a B cell that does recognize spike, guess what? That labeled spike sits to the surface of the B cell. So we've now got a B cell which is fluorescing green and red. So then we can use what we call flow cytometry to separate out those cells. So we ask the machine, give us all of the cells which are fluorescing green and red, but give them us one at a time, because we can't cope with multiple ones. We can, but we have to do a different thing. And then put them into an individual well of a microtiter plate, so it's 96 little wells. So how does it do it? T cell, don't want that. Don't want the T cell. Here's a B cell we want, so it sticks it into that cell. T cell we don't want, rubbish. B cell, not the right sort, it's not labelled. T cell again, T cell, we don't want those, but we want that labelled B cell. And so we go on, and so we end up till we've exhausted that sample and sorted all of the spike reactive B cells. So once we've done that, we can lyse that B cell and use molecular biology to amplify the genes which encode for the antibody that we're sitting in the cell. So we can literally, it's going to take an hour to amplify the gene that encodes for the variable part of what we call the heavy chain and the variable part of the light chain, and together these form the antibody. So we end up with 2 bits of DNA, one that encodes the heavy chain, one that encodes the light chain. Guess what? We can put them in the cells that we usually use, and once it's in the cell, they'll produce the antibody. So this time, rather than producing virus, we're producing antibody. That antibody gets secreted into the media that the cells grow in, the liquid that the cells grow in, and then we can screen the antibodies that each of those B cells was producing. So that then we can say, right, hopefully here's a spike reactive antibody, and that antibody kills virus. That's the theory. So does it work? I've got 50 seconds left. Can I do it? Probably not. I'm going to run 2 minutes over. Is that okay, Marie? All right. So what we ended up with is producing a panel of antibodies. So these were antibodies that didn't have that elongated, loopy CDR3, but we were mainly interested in these antibodies, which did produce that really long, extended CDR3. Green, again, is good. Here we've got different versions of virus. You can see different versions of the Wuhan and the original virus, and then we have alpha, beta, and Omicron variants. And you can see that the reactivity of the antibodies which aren't ultra long is generally not great. There's this one antibody here which has got reasonable cross-reactivity, but some of the others lose binding at all to some of these variants. So they're no good as a therapeutic for the next coronavirus outbreak. But hopefully you can see here for the ultra-longs, we've got a number of antibodies which maintain that cross-reactivity against all of the variants that we throw at it. But binding doesn't mean does it kill the virus. So then what we do is use the same antibodies in a virus-killing assay. So we have the pseudovirus assay, you know, they're using the HIV as a kind of surrogate. And you can see here that P7, which was this, whoops. So this antibody here, I'm just showing you the data for, if I can get this to work. I'm getting panicky here. Here we are. So that one there. So when we test that, we can see that it has good neutralization. So the virus is killed, and you can see that it acts against all of these different variants, including the original SARS coronavirus that this cow never saw. So this is going to be like the other SARS-BECO viruses that we're hoping the antibodies will be active against. And if we take it with a live virus, so this isn't the surrogate, this is a real live SARS coronavirus, we see the same kind of activity. So that we know these antibodies not only cross-react, but they also kill the different viruses. And then finally, does it really have pan-SARS-BECO virus activity? Does it really recognize viruses that we take from different bats with the same kind of reactivity that we see with SARS? And hopefully you can see here with MAB7, you get very good reactivity against all these different variants of the SARS-BECO viruses. All of these are still loitering out there in bats. So if we get a spillover event from one of these, then hopefully we've got an antibody that's ready to go. It also protects hamsters, so if you infect a hamster, it doesn't go, choo, it loses weight apparently. Rich, you'll have to tell me, yes? Loses weight, you can also measure viral load in the nose and in the lungs. And you can see here, for example, here's the antibody that we're interested in. These are mice that haven't had any antibody. This is the viral load in the nose, and you can see our antibody reduces that viral load. Again, in the lungs, which is more important, because it's the pneumonia that we're interested in. Again, our antibody's very good at reducing the viral load. Much better than a currently licensed antibody, which is used, I think it's the only one still in use for the UK. I think in the US they've got rid of pretty much all of them. Okay, and for the anoraks out there, it's always good to understand how these antibodies work, because sometimes that can lead you towards vaccine design. So you can start to do rational vaccine design. So here we can see different antibodies. So this is antibody 99, and how it attaches to that trimer of molecules. We can see that each of the antibodies locates within the receptor binding site. When we tried to resolve the structure for that antibody that we're most interested in, we couldn't resolve it. Basically what happens is, as soon as the antibody binds, the virus falls apart. So we just couldn't get the structure. But it gives us a clue as to how it works. So I will finish with a bit of Lewis Carroll, and that's essentially that basically every day we are in an arms race with viruses. The virus constantly evolving, we constantly evolving, whether it's our immunity or whether we physically are evolving. If I had another hour, I'll tell you all about HIV and their endogenous retroviruses, and the fact that you're only sitting here today because of a historic infection of a HIV-like virus, which gave you the ability, not everybody in this room, some of us, the ability to form a placenta. Because without that previous endogenous infection, mammals wouldn't be here. So, you know, we have been evolving and counter-evolving with viruses in very much the same that we have. And it will go on forever. But I don't just do virology. Back to the building. I do love building. These are my two last building projects. That was a shed I built for my son, who now we never see, which is a result. Okay, that's my advice: build a shed. Okay, and a pizza oven. That was really fun, because essentially you're employing kind of ancient Roman building techniques with a tremolo arm. And anyway, I'll leave it at that. Just to really finish, to acknowledge all these people, there's so many people that I've probably forgot. I'll have to mention Rich because he's only in the audience. Otherwise, nada. Okay, and also final acknowledgements to my PhD supervisor, Ulrich Dasselberger, who I didn't follow to Cambridge, but he's been supportive throughout my career and allowed me to do things that maybe lots of supervisors wouldn't have given me the freedom to do. My family, my wife, Dawn, Rachel, my daughter, Peter, my son, and of course, Billy the dog. Thank you.